Welcome to the Grand Rounds for the Center for Healthcare Innovation. And it's a great pleasure for me. I'm Terry Clausen, the Academic Director for the Center for Healthcare Innovation, to introduce this morning's or afternoon speaker. It's not here in his bio, but my recollection is Dr. Ryan Zarychanski arrived back here in Manitoba, University of Manitoba, about the time I was coming back to Manitoba. I came back in 2010. He might have come back in 2009. I'm not certain. But when I came back, Dr. Cheryl Greenberg, who was my department head, was telling me about this really bright, great clinician scientist that had just been recruited here. So that's how I got to learn about Ryan Zarychanski. And certainly he's been a great part of making my 9 years here at University of Manitoba interesting and stimulating and everything else. So who is Ryan Zarychanski, you may ask? And he didn't want me to focus just on all the academic details, but we do have to do some of that. He's currently associate professor in the Department of Internal Medicine, the section of Hematology, Medical Oncology and Critical Care, and the Department of Community Health Sciences here at the University of Manitoba. He's a hematologist, critical care physician, and a clinician scientist here, both at U of M and cancer care. His research focuses on hematological aspects of critical illness, and he has a major program looking at unfractionated heparin in septic shock, where he travels around the world. Brazil, I never know when I get a text from him which country he's in, so it's always an important part of our conversation. And so he has a major research program. I think more recently, and probably what led to this rounds in, was his study with a group of his colleagues here, that he developed a grant proposal on rewarding success, which really is looking to transform how data is used in clinical practice and research. And he's really leading this provincial effort to, in a collaborative context, to develop an integrated real-time clinical data platform. But I must say, from a personal point of view, I've always enjoyed, we spend many a lunch at Halong Bay. This is not a commercial announcement, but he knows what to order. It's always a good time. I've seen him as a father. He's very proud of his two kids, and he never seems happier than when he spends time as a husband and father at their wonderful cottage at West Hawk Lake. I think that's an important part of his life, but he still finds time to work pretty hard and drive forward the agenda here. So Ryan, I'm really looking forward to what you have to present to us in the next whatever time you have. Thanks.
Thank you, Terry. That was really kind of you. Terry's been a great inspiration for the work I do here, and when I think that things can't be done, Terry's the one who always says, keep going, you'll get it done, and it is possible, and so he's a great visionary, and thank you for that mentorship, Terry. Okay, so I was going to talk about what we do now, but it may not be what Manitoba does right now. But I hope that we get there in short order with many of the collaborations and the opportunities and the great people we have in the province. So I was going to talk about so looking forward to the present, or what might be going on in other places to some extent, but really it's what's around the corner and what we have not missed the boat on, but what might be coming that we could adopt. So I'm going to talk about some of the challenges with current trial designs. I'm going to have a bit of an RCT lens, I have to say. It's not all I do, but I wanted to make it a focus of this presentation. Some of the opportunities for innovative trial designs and research methods, but maybe a bit of a vision for creating a learning healthcare system in Manitoba. Okay, where this all started, I think this is where it may have started. James Lind. So he was like an army physician in the British Navy, I think, and people were dying doing the journeys across the Atlantic, I guess in other places too, of scurvy primarily. And they didn't know what caused scurvy at the time, but he thought it might be a deficiency in what was going on in their diet, but wasn't quite certain. So to make an experiment, James divided up 6 groups of sailors. There's 2 in each group. He had the foresight to have a standard of care arm. Do I have a? I don't have a. Oh, yeah, right there. I have a standard of care arm. He gave some nutmeg and garlic, some diluted sulfuric acid. I'm not sure I wanted to be in that group. cider, seawater, and oranges and lemons. And what did he find? He found out that if you provide the oranges and lemons to this cohort, they did not die or get very, very sick of related vitamin C deficiency. There's a lot of great stuff here because it brought in the concept of causal inference, distributing groups separately to understand what treatment effect would it specifically have on a person, and it changed the way the Navy did their daily business. They all of a sudden had fresh fruits, citrus fruits, particularly lemon, to prevent this disease. There are some things that weren't done all that well too, but the fact that he had standard of care arm was fantastic. There was probably no consent. There was no patient engagement. There was extreme comparisons. Like, I'm not sure you could give acid to someone and think that group is going to do well, but I'm not sure what he was thinking. So there's an extreme comparison that we try to avoid in practice. The patients were essentially non-randomized too, and so there might have been selection bias, thinking, well, you're kind of sickly anyway, and I'll give you the acid, and so that may have gone on there. But it changed practice and became a template for how we might do research. We've improved upon that since that time, and I'll go over some of those. I think really interesting, this easy experiment done on sailors was published. This is the Treaty on Scurvy in 3 parts. So duplicate publication existed in 1700s. It wasn't just us trying to publish our stuff more than once. It was James Lind at that time too. 3 parts just to show that a bit of lemons worked. And so there are great strengths to the parallel group, or the several parallel group RCTs and trial methods right now. And I'm going to go over some of these. Like, it allows you to isolate exactly the experimental variable of interest. Like, you break up into different groups, and you're changing only one thing in each of the groups. These RCTs have very high internal validity, and if they're done well in large sample sizes, in a high quality, they should have low risk of bias and confounding. And even if you miss a few confounders, in a large randomized control trial, the unknown confounders should be equally distributed or non-differential amongst the groups. And that is a difference from when observational data. This idea of randomization where you don't have a choice, a flip of the coin in the past, the computer in the present, decides what group will be allocated to each, can really mitigate or hopefully abrogate these confounders, and the truth, hopefully, at that time, knowing that truth is iterative, will be hopefully revealed. There's accepted statistical theories, the way we analyze RCTs, and there's a robust ethical framework now that really occurred after the World Wars to protect patients who are engaging in research. But there's legitimate challenges too. Randomized trials are expensive and difficult to fund. In every cycle of CIHR, our National Granting Funding Agency, we can only fund about 7 or 8 large, well vetted and high quality randomized control trials. But there's a ton of questions to ask in medicine, and without this evidence, this high level of evidence, we could be providing expensive treatments or therapies that may not work the way we think they may work. They're expensive. They can sometimes have marginal generalizability, sometimes not, but if they're highly explanatory to show that does something work or not specifically in a mechanistic fashion, it may not apply to everyone in the population, just under these highly experimental conditions. But I'll come back to that we tried to avoid. They also, their outcomes in RCTs are typically short, and so they might help you survive hospital admission, but are you still alive in 30 days? Are you still alive in 60 days or 90 days? Sometimes follow-up RCTs isn't long enough. And they have a long cycle of research. It could take 10 years, not to do one, but to provide the background data, to get the preparatory grants, to then move to the next phase, and then eventually have a phase 3 trial and report on the results. It could be 10 years, and then another 10 years or worse to get it into practice. So the research cycle of providing this high level of evidence is unfortunately long. They generally test one hypothesis at a time. They're not efficient for rare conditions, so they're not usually the right treatment, the right study actually. And you have to ramp them up to get the question answered, and then they come down, and then you got to ramp it up again to answer the next question that you're interested in, or that are hopefully a priority to patients and families. The statistical theories are generally around frequentist approaches as opposed to Bayesian approaches, and it requires some predicting of what the effect estimate is and the sample size from the beginning based on hopefully preparatory work, and then seeing that through to the end and hopefully being right around the sample size that you chose or the effect estimate that you guessed upon. And so a lot of your investment is unfortunately contingent upon not your best guess, because that makes it seem like it's not informed, but your best guess. And sometimes you're right, and then you've got a well-powered study, and sometimes you were wrong about the baseline effect rate, whatever it's mortality or transfusion or whatever, suicide, cancer recurrence, and sometimes you were wrong. Sometimes your effect size you were right on, and sometimes you were surprised, and you were overpowered or underpowered, meaning that it took more time, less time, spent too much money, didn't spend enough money. And the how we interpret these results is heavily dependent on the p-value, this concept that's entrenched in frequentist statistics, where something is significant or not, it's dichotomized as important or not, and that's a problem. And here's a question that I'm going to ask the group, and you'll get to respond. And will it be obvious on how to respond from the question? It comes up on the top. Oh yeah, it doesn't come up on my screen, so that's good. Okay, so so on your smartphones, if you go to a text option, like you're going to send a text, and then bring up a new text and text this number. Like, this is not the content, this is the number you're texting to. You're texting to 37607. That's the number you're going to text to. And then in the message, what you want to send to 37607 is CHIMB622, okay? That's the message. So do that, and then you'll be activated on the Poll Everywhere system. So it's like a Jets game, I think. And so once you're activated, you'll be able to answer A, B, or C, or D, I presume. A, B, C, or D. I'm new at this, and I'm a guinea pig. So there's an RCT, a randomized controlled trial performed that compared drug A versus a placebo. I guess there's no good standard of care right now, so placebo is ethically acceptable. The results show that the mean arterial blood pressure, or the MAP, for the drug A is lower than placebo, assuming blood pressure is a good thing to lower, okay? And so it lowers it by some amount. And the p-value is 0.04, okay? How should you interpret this p-value is the question. So A, do they text A or B or C or D? I think so. That's what we did in the practice. So if you think it's A, then do this. So there is a 96% chance that drug A lowers MAP more than placebo. If you think that's the answer, do A. If there's based on that p-value 0.04, is there a 4% chance that drug A is no different than placebo on its effect on the blood pressure? Type B. And if you think neither of these are correct because I haven't given you the right answer, which is totally possible, then pick C. And if you think both are right, it's just a different way of looking at it, then do D. Check this. So try do something, even if you're not sure. Do something. I don't have the ability to do anything on my screen. So can you? Yeah. Like there's no.
So you have 24 responses on the right hand?
Oh yeah. So I don't see any of that on my presenter view.
I can show the responses when you're ready.
I kind of regret being on presenter view. Okay. So there's more than 27 here, so some of you haven't answered. We will wait. Okay, so let's see how, so any time is good, I think. Neither. So most people did not interpret this correctly, for sure. I tried to simplify the answer, and the most correct answer, although if Alan's here, he'll take issue with it a little bit, but or Rob. I see Rob. I'll take you. So forgive me, but I tried to simplify it a little bit. And really, we're talking about null hypothesis testing with frequentist approaches. And so what we're saying is that there is maybe a 4% chance that if these are that the drugs are equal, and there's a 4% chance that they're actually not equal. So it's rejecting the null hypothesis. If we repeated the study many times in the same population even, then we were more likely to not find the null hypothesis. That's pretty backwards, eh? And that's how we test in medicine. It doesn't make sense. We're not showing that something is better. We're showing that based on some probability, in this case 4%, the drug there's a chance that and we think it's significant chance that they're not equal. So most people don't know how to interpret a p-value, and this group reflects that. It's okay though, don't worry, it's not a judgment. Most people don't know how to assess a p-value. And this is what we're faced with every day, is this dichotomous, is it significant or not, based on null hypothesis testing, where we calculate the probability of something is as big or bigger than not being equal. And so if it's larger than some effect that we don't think is, that we think is important enough, then we say it's significant. The problem is we apply the same important enoughness to every clinical situation, whether it's mortality or hangnail removal or suicide or cancer recurrence even. And that's another problem, is that our significance threshold is constant when our judgments and our willingness to accept the outcome is clearly not equal. So we have problems with trial design. We have challenges with statistical theory. Our methods of consent were made up over 70 years ago, and they equally have not dramatically changed. They've been tweaked a little bit, but they've been mostly a one-size-fits-all. Someone comes in, they are going to get involved in research now because we understand research on prisoners and research about their people's knowledge is not a good thing. We want to respect their autonomy, so they know that they have a choice and they're informed about the research experiment and they can participate or not, understanding the risks and benefits to them. And we've applied this one-size-fits-all model of informed consent where we have like a 10, 20, 30-page consent form filled with all sorts of legalese to protect lawyers under the guise of informing patients, to protect institutions at the request of the lawyers. It's not the lawyer's fault. And the patient has been increasingly forgotten in the process. And the concern has been, I think, more at minimizing legal risk than really informing patients. And the methods of consent have been incognizant, or they haven't really considered risk. The risk of me dying from an unknown medication is me different than the risk of knowing which accepted medication that's currently available in practice that are used equally every day without my consent is better. Right? That sounds like if there's 2 antibiotics and we reuse them every day and we just don't know which one has better outcomes, that risk is different than there's a new cancer medication. I'm not sure what it does. Most rats live, but some didn't. But and I think I should let you know about this. That's a different kind of experiment. We historically have done research without patients. The doctors knew what questions to ask. James Lind knew what groups they wanted to choose, what was important to him. But patients there's a good sense of priorities. They pay for the healthcare system. Their tolerance for risk under certain situations are different. The methods of consent that have been used on them haven't been well studied to some degree, and I'll come back to that with our patient engagement experience. The trial outcomes that are important to patients haven't always been what's important to them. Like for a lot of times we studied a drug medication that I presented on a few years ago that increased blood pressure in people with dangerously low blood pressures, and it turns out the medication does increase your blood pressure, and we used it based on that. But then when we looked at it further, it killed people. And that wasn't a great example of that. The patients would have preferred to know if they lived or died, because they would have, and that's obvious. But the physicians didn't even know or didn't study the right outcomes that were always important. And patients have contributed vastly to outcomes in orthopedic surgery, in cancer, mental health that have been important to them that have been overlooked by physicians historically. Okay, so how do we move forward from our challenges? I was just in China, and I was in the washroom. and there's urinals in men's washrooms, and some people, they don't move forward enough towards the urinal, and then they make a mess on the floor. And in Chinese, it said, one small step forward, one big step for civilization. And reminded people how to use the urinal, I guess, in a men's washroom. Anyway, so how do we move forward to advance our trials methodology and overcome the limitations we have in current research methods? I'm going to just in a cursory fashion touch on some of the trial designs beyond a randomized controlled trial. I'm not going to talk about the first two because they're actually not that innovative and been used historically for a long time. Factorial trials and non-inferiority trials. And factorial trials study more than one intervention at the same time, looking for interaction and treatment effects. And non-inferiority trials study something that is a bit of a different question than we normally do, which is which one's better. It has a bit of a different slant, which is which one is not any worse than something else. And those, I'm just going to put that on the screen but move forward to something more, I think, more relevant to where we're going now, which is pragmatic trials. So in the concept of randomization, we take patients and we allocate them by chance to different treatment groups, and that's called a paralog group RCT. Usually there's 2 groups, sometimes there's 3, sometimes there's more. When there are more, they're messy, so usually there's 2. And I wanted to touch again on pragmatic trials. Pragmatic trials are trials done under usual conditions. And again, this is something beyond the standard RCT that has limited generalizability, that may not apply to my patient, that takes a long time to conduct. And using pragmatic research techniques, one might overcome some of these limitations. And in a pragmatic trial, we study interventions that are used every day under their usual conditions, how we typically dose the antibiotic, regardless of all the variability out there. We might say, okay, let's accept that variability and just say, if you give that drug versus not giving that drug under usual conditions, what's the outcome? This concept provides a much more generalizable result and impacts practice. That changes it because people see the result and say, okay, well, I use that drug too. If I continued to use it as studied, I'll see this effect. If I give it up or not use it, I might see this average treatment effect. This is if you're going to do trials and think about incorporating pragmatic aspects versus highly explanatory aspects, which is under this specific laboratory situation with very strict inclusion and exclusion criteria. If I give this drug in this one dose, what is the short-term treatment effect on this physiologic variable? That's highly explanatory, which you can see that may not apply well to the patient's bedside. And so these are called Precy 2 wheels, where they score the pragmatic aspects of many things, like the eligibility criteria, the recruitment, how you use the drug, the follow-up, the whole all the concepts of pragmatism. And look at even the question that's asked, what is the postprandial anti-inflammatory effects of extra virgin olive oil? This is after you eat the oil, well, how do your inflammatory markers change? Do you care about that as a patient? Probably not. You'd care about, well, if my inflammatory markers change, what is the outcome to me? What's the clinical expectation? I might have less heart disease. I might have less autoimmune disease, something like this. And so this is a highly explanatory trial that doesn't have a lot of clear applicability to a patient versus something like this. If I'm a woman and I have an uncomplicated urinary tract infection, do I need antibiotics or can I just take anti-inflammatory medications? And the truth is they both are effective under the situation. And this is under usual care situations where you recruit people from practice, you have very little exclusion criteria other than uncomplicated UTI. You give antibiotics in whatever dose that they typically prescribe or NSAIDs, that's ibuprofen, and you look at the clinically relevant outcomes to that patient group. So a highly explanatory versus a highly pragmatic trial. This is a trial that we're doing right now with myself and Brett Houston, who might be here. She's a PhD hematologist, and we're evaluating a drug, a very inexpensive drug that reduces bleeding in specific surgical populations. And what Brett is doing is she's organizing with other co-investigators a large pragmatic trial where she's testing if there's a treatment effect of using this drug under the average conditions based on preparatory work to a large group of surgical population group that have high risk of bleeding, to see if we just give the drug en masse, if we can reduce transfusion to a large extent, therefore saving money to the healthcare system and preserving the blood supply. And we're getting the patients from the OR as they're booked. We're giving the drug normally as it would be used in practice under the standard conditions. We're giving the drug as the anesthetist would give it right now. We're not even following the patients up. We're using a data-driven solution, so we don't even have to go back to the patients and ask them for their outcomes or collect it that way. And we're analyzing this by very pragmatic methods. And so and the idea of that trial is that it will hopefully change the world. And people will say, okay, well, that's such a generalizable study in 6,000 patients, if I just give this drug or not, that all healthcare systems hopefully throughout the world will adopt the therapy if it's shown to be beneficial. So beyond pragmatic trials, I just wanted to touch on some other things: registry-based trials, cluster trials, and adaptive trials. Registry-based trials is what I described. And so what's the most expensive part of conducting a randomized control trial? I didn't ask this as a question, but can anyone offer a guess? The most expensive part of a randomized control trial? Staff, personnel. And what are they doing? Collecting data. And a lot of data is already collected in the healthcare system. Whether you went to your doctors or not is definitely collected. Admitted to the hospital or not, went to the ICU, got certain drugs, had a procedure, was discharged alive, were alive in 30 days, got outpatient medications, etc. A lot of these hard outcome data is already collected in computers, but for randomized control trials, we recollect them. And registry-based trials harnesses all the data that's already in our multiple computer systems and electronic health records to generate the outcomes of interest for a randomized trial, reducing the cost by 50 to 70%. This is an example called the TASTE trial, which was with the first large registry trial done in Sweden, where people came in with a heart attack, and they were just randomized to one intervention or another, and no data was collected. And they enrolled 7,500 patients almost. They had no losses to follow up because the data was all driven by the healthcare system itself. They showed that there was no difference in two different therapies that were used routinely but had different expenses. And at the end of the day, they stuck with using methods to open up the artery instead of retrieving the clot that is present in a heart attack. And so it defined the best standard of care at a fraction of the cost quickly, because every patient was essentially in the study, and inexpensively because the data was collected already by the healthcare system. The Traction trial that Brett and myself and others are helping to organize is also using this method, where we're studying the effect of a drug versus usual care in a waive consent fashion, because the risks to the patient are very low. The drug is used every day in practice and approved as such. And we're studying it in a group of about 6,000 patients in a trial that costs we think about $1.25 million. A typical trial at this size, using standard consent methodology and standard data collection methodology, would be $5 to $6 million. And maybe unfundable in the Canadian grant funded context. So that's parallel group trials. Cluster trials are a little bit different, where in the past, in the parallel group RCTs, we randomized individual patients. Each of you could be split in half, and half of you would get one drug, half of you would get another drug. The concept of cluster randomization is that the unit of analysis and randomization is a group of individuals, and it could be schools, it could be communities, it could be cities, it could be clinics, and this is an appealing study design when asking questions at a policy level. Like if I universally bring in this to my school, like HPV vaccination, to my clinic, like some kind of hand washing precautions, or a staffing model, what will be the outcome to that group of patients, not to the individual now, to the group that's studied. It can often be more generalizable, but the results apply at that cluster level, not at an individual level. And just as an example here, like where you have clusters, let's say they're schools, and schools might be like all, if this is racial, all one race, a mixed race, a little bit more of something, a little bit more of something else. And those groups are randomly distributed now, but at a school level, to one group or another group. And some intervention is applied. In these trials, the sample size very much depends on the number of clusters, in fact, and very powerful compared to even the size of the clusters. How you interpret them depends on how similar they are and how the treatment effects vary among the clusters, and we call that the interclass correlation. And they're not as good, like you need more people in general to study than to find the same power of therapy, or power of statistical power than a parallel group RCT, so you often need more patients. But that is generally a trade-off of it's easier to conduct because we're enrolling whole schools now, or whole clinics, or whole hospitals. And so you trade this off. They often are very good to study low risk interventions where individual patient consent is not required or appropriate. Beyond that, there's something called the cluster crossover trial, which I'll give an example of, where now you're randomizing the clusters, Group A, Group B, but then you switch, where Group A now gets Group B for a while, and then you switch back, and then maybe you end there, or keep switching throughout the trial, and that increases the statistical efficiency often, depending on some of this clustering effects. And so each cluster becomes their own control. An example of this is something we're doing right now. And so this isn't like some pie in the sky research technique. This is what is being done right now in our hospital. In fact, 8 or 12 hospitals around Canada, called the B3 trial, where they're studying benzodiazepines. It's like an anxiolytic kind of anesthetic drug in cardiac surgery. So they're studying the effect of using it or not using it, led by Jessica Spence and Eric Jacobson from Winnipeg. And they're evaluating using benzos or not at a hospital level, a policy of using benzos at anesthetic induction or not. And they're doing this in a cluster crossover trial. The traction trial is that I presented about the tranexamic acid is also being planned as a cluster crossover trial in Canada. This is a step wedge cluster design that I'll talk about next, which is a kind of a cluster trial again, but in this trial design, the intervention is brought in at different random points to different hospitals. So here's an example of a trial that we're doing right now called the Stratus Trial. We're studying if low volume tubes that you collect blood from a patient, if you change the volume of tubes, you can significantly change the transfusion rate of patients. Because if you take blood away for diagnostic testing, then it might make them anemic, and then they need more blood transfusion. And so these are all the hospital sites that are participating, and this is when they're in the control arm here, and then based on, this is random, so it's not 1 to 14 or 16 that's participating, 16. Randomly, the sites get assigned to when they cross over to the low tubes. So this is the step wedge, and that's called the step. And eventually, all sites will be using the new tubes. And so this kind of trial design is really good for bringing in policy decisions, but particularly a policy where you kind of don't want to ever go back, where you think that once everyone is using the things, using the new intervention, assuming that it works, you'll stick with it because they never crossed back. In addition, if you think there's a large carryover effects, if you're using one drug and then switch to the other, and then the other drug carries over to the new group, likewise the step wedge might be a good design for that. And so in this trial design, we're studying over like in 10,000 patients, a very small difference in transfusion amongst individuals from a mean of 2 units to 1.94, which doesn't sound like a lot, does it? This is like a fraction of a unit of blood. But on a population level, the size of Winnipeg, the size of Manitoba, the size of Canada, the size of the world, you could save a lot of blood. I'm lastly going to just finish this part of the trial design methods that are being, again, used right now in practice with adaptive trials. When I started the talk, I mentioned frequentist approaches where we have to guess at the sample size, the effect size, everything at the beginning, and then we let this play out over the course of 2,000, 3,000, 10,000 patients, $1, $6, $10, $20 million, and hope that all our guesses were right. And the truth is we're not always right, and we waste a lot of money and a lot of time and resources. And there are methods now available to be more adaptive, that is, to increase the flexibility of the trial by using the information that you collect along the way of doing the trial. They can be more efficient, more informative, and more ethical, and I'll show you why. They can definitely make better use of resources, and there's several adaptations that exist. Like you could refine the sample size with information available to you as the trial accrues. You might even abandon a trial. If there's like 3 arms and one of them is not very effective compared to the usual care, you might abandon it. Likewise, if one group is more effective, you might weight the randomization to that group so more patients are receiving the more effective therapy. That's the more ethical part. You might even change the ratio of the amount of patients, I said, randomized to the arms. And you might stop the whole trial early. You might continue it further based on your sample, based on your effect size that you see. So you're making adaptations along the way. We are participating in one of these an adaptive trial. It's my own trial, and it's a small adaptation, but I hope that an important one in a drug that we're studying called heparin. Heparin is a blood thinner that's used for thinning the blood and in many conditions. But we're studying it as an anti-inflammatory agent with myself, Dr. Houston, who's in the room here, and Anand Kumar, Dean Ferguson, and people from around Canada and the world. And it's a worthless drug that we think could save maybe 4% of people's lives, a reduction of mortality in 4%, that is, in people with severe infection. But there's no money to be made in it, and it's not very sexy. And so it's been hard to get funding for this drug. In particular, it's been all taxpayer funded. And so I could guess the treatment effect that I'm going to see and ask for $5 million and maybe get it or maybe not. But what we thought we would do for information, for social responsibility, use of your money most efficiently, we are going to randomize 500 patients first and see the average treatment effect at that time, understanding the probability of having enough sample size based on an effect size that we still have guessed. That's one way we think we're going to do it. The other way is to go full Bayesian, which has changed from frequentist to Bayesian methods to really inform this most efficiently and continuously as we can, so we have the right sample size to fit the right effect size that we're seeing as the trial unfolds. This trial is going on in 7 countries now, and we just, like last week, had our investigators meeting in Brazil where we started up 15 sites there, and they're going to participate in enrolling the first 500 patients in this adaptive trial. And the phase 3 trial sample size will be based on the observed treatment effect, the variability, follow-up, attrition, etc. This is a study design called an adaptive trial, an adaptive platform trial, where it combines elements that I've talked about in adaptations onto a platform for a trial that never ends. And the idea here is to study different treatments, different patient populations, different effects at the same time. It allows you to keep a trial going so you're not taking down that infrastructure, rebuilding it, and pulling back therapies that are ineffective when you know the probability of them being ineffective is such that it's obvious from the analysis. And likewise, adopting a treatment as routine when the probability of it being better than what you're comparing, again, is obvious. And the opportunity here is to build a learning hospital or healthcare environment where every patient is essentially enrolled in a trial that doesn't end, and it's perpetual learning where every patient informs the next, and every success is then is adopted into therapy, and every failure is de-adopted. And we're doing this around the world right now, being led by John Marshall from Canada. I'm part of a grant called REMAP-CAP, where we're studying different therapies in acute respiratory failure. And so these are high mortality populations. And this is an example where we're studying different doses of antibiotics here. And this is a hypothetical situation where this is how a treatment arm does, how a treatment arm gets randomized, and how this low-dose antibiotic doesn't do so well in the adaptive trial. So eventually we phase it out. This medium-dose antibiotic is doing really well, so people get allocated more as the trial progresses. Eventually everyone's on the antibiotic, and then we start the new therapy. This is done by full Bayesian methods, and I won't get into all that in the context of this round, but I just wanted to just communicate that this adaptive platform is something that is more than actionable. Is it fundable? That's hard. Terry is leading a group of pediatric investigators to look at 4 very innovative trials in a platform situation as well, with some important changes though, whereby we're both struggling about, well, after the first trial is done, after the first answer is, how do you keep getting funding? How do you sell the concept of a trial that never ends to a funder or to a hospital? Until there's a cultural change where we really want to integrate research into clinical practice, that might be a challenge. Okay, I will skip that, and this talks about sort of the Bayesian methods, where if you're uncertain if a therapy in this trial works or not, with enrolling one patient, this could be your probability distribution of something working or not. If the trial, if the patient has a better outcome because they were randomized to the drug that they received, well then the probability distribution of them surviving or whatever it is might go up. It might go up further if the next person benefits, and then it might tank if the next patient gets too randomized to the drug, has a bad outcome. And eventually your outcomes accrue over the trial, and you develop a distribution of you surviving or not. This is the outcome of survival here. And you compare that to what you thought their survival ought to have been from previous studies or from known literature. And then you're comparing these things to see, is this drug really different or not? Beyond everything that I've talked about, there's a whole introduction into medicine about omics and genetic data, personalized medicine, that is an absolute other talk and very difficult statistical concepts to understand and to communicate and to really bring into practice, where the p-value fails horrifically because it's so dependent on sample size. If you have a sample size of 1,000, then you're more likely to show a statistical benefit if the treatment effect is small. If you have a sample size of 1 million, or 1 billion, then you're more likely to show significant treatment effects. In fact, you will show treatment, demonstrating anything. And this has been shown in drug registry data, which kind of goes to the next thing about big data, where you have, if your sample size is large enough, I think this was a Swedish or UK study, that they could demonstrate that 3/4 of drugs cause cancer or are associated with cancer. 3/4 of drugs we use are associated with cancer. Because the treatment effects are small, they're confounded, and because the sample size is so big, small differences are statistically significant, which of course is not in anyone's interest, and we have to figure out how best to reconcile these issues. You'll hear more about artificial intelligence and machine learning. We're still really getting used to techniques in that, and we haven't, we're exploring how best to incorporate those and understand the results. I'm going to finish just with, I talked about new trial designs. I just want to talk about innovative methods of consent. I mentioned earlier that the methods of consent are old. They've been really unchangeable. They benefit institutions more than patients. And if we're doing trials in the 21st century, which we are, not only are we considering new trial methods, but we are considering and incorporating and using new methods of consent, such as deferred consent, where you might start the drug and then talk to patients later if it's an emergency or people aren't available and their lives are at risk. They might have, there's concepts called integrated consent or wave consent. And you're using all of these in trials now based on patient preference, based on risk to patients, urgency, impracticability, importance of the treatment answer. And all these have a very important role to move research beyond the traditional informed consent that one size does not fit all, as we know. Likewise, sort of a novel concept that Kate and others at CHI have been really championing are the idea of patient engagement, where the doctors and the healthcare team made all the decisions before, didn't engage patients, may have got it right, frequently didn't understand their priorities, may not have had the right outcomes, may have though. And patients now have an increasing role as partners in research. We've incorporated patients in all our research that we've talked about, the HALO trial, studying heparin, traction, studying tranexamic acid, and others, where patients are involved in picking the right study design, understanding the methods of consent that are most appropriate and acceptable to them, having input into the study materials. Like an example is that we had like a 20-page consent form that was radically changed to 6 pages based on patient input. That was really a negotiation because the patients wanted it 1 page, but the ethics board demanded core content that was 6 pages, even though the patients thought it was not in their best interest to have that material. So ethics boards are willing to listen, but we're still finding a way to really incorporate the patient voice, protect autonomy, meet institutional requirements. This is Mac, who's the chair of one of our patient engagement groups for one of our sepsis studies that coordinates a lot of these activities for patients, but then is part of our steering committee as a patient partner. Here's an example, and the next question. If you were offered a blood thinner to prevent stroke, so you're a patient, you're at high risk of stroke. This is a phone question. And you were offered and suggested that you be on a blood thinner to prevent a stroke. That's when there's a blood clot in your brain, and then you have some neurologic disability because of the clot in your brain. How many bleeding episodes would you accept, because it thins your blood? How many bleeding episodes would you accept to reduce your risk of a stroke? There's no right answer here, by the way. It's just more of what would people think. And so would you accept 1 bleed to prevent 1 stroke? Would you say, I'm okay if I have 2 bleeds as long as I don't stroke? Would you say, like, I don't want to stroke, 5 bleeds I'm fine with, 10 bleeds is even fine because I really don't want that stroke? What would you say if you had to start on a blood thinner? I hope that you would want a bit more information, like, well, what's my risk of stroke? Like, how bad are these bleeds? You know, like these are important questions. You know, what's the outcome of a bleed compared to if I stroke? But you don't know these right now. And so most people would say that they're willing to accept some bleeds to prevent strokes. And it varies according to patients without even giving you all the information. And that's what we would find. And what we found in our studies with incorporating patients in this decision-making process is that patients, when given all the information that I didn't even tell you, are much more willing to accept a bleed risk than their doctors. The doctors will withhold the anticoagulation, thinking that I don't want them to bleed. But the patients are saying, I don't want a stroke, man. Like, give me the drug. And so it's an example of where patients have really informed practice by incorporating their concepts of risks and knowing that we have to have this discussion, or we might make decisions that don't really reflect their needs, their expectations, and their fears. What about big data? You'll hear more and more about big data in 2019 because we have the technological ability to connect electronic health records, to connect us all with social media, with Fitbits, whoever has them on, and all our data that's out there. And the statistical methods to analyze these observational studies, essentially they're just evolving observational studies of people doing things, using stuff, buying crap on the internet. will there be a need for a randomized trial after this? All of these big data are so interconnected that we can really tell everything about us by just you harnessing the technology in front of us. And so here's a question for you: is that this? So using data from 15 U.S. advertising experiments at Facebook, comprising 500 million user experiment observations, 1.6 billion ads impressions, they compared the ability to predict use, like buying and behavior based on ads, based on two scenarios. One, if they just followed the data and followed the use of the ads that were shown and what people did now with regard to their behavior, like that is usually buying or seeing or visiting, versus if Facebook randomized a person to receive a certain ad or not receive an ad on a huge scale, okay? And so Terry reminded me of this study. Thank you for that. And so how do you think, using sophisticated analytics and large data sets, how do you think these compared? So using the observational methods, these methods were able to predict more accurate treatment effects at considerably less expense than the RCTs. So a bit more accuracy, less expensive. The observational methods generally produce the same effects as the randomized experiments, because they're, the techniques are so sophisticated now, they can account for all these confounders in robust methods, or the observational data failed to produce the same effects as randomized experiments, even after trying to account for the multitude of confounding influences. How do you think those fared? Yeah, you're right. So C is right, is that the observational effects were different than the randomized control trials. The first one is irrelevant because we don't know what the truth is, so the accurate prediction is unknown. Maybe and the confidence limits were probably the same because the sample sizes were essentially the same with that large group of people. The costs were very similar because the data is already collected and it's just a computer randomization of whether you got one ad or the other, and that's where we're going now with innovative techniques. Randomization is cheaper. It can still be, it is powerful, and you don't get the same treatment effects sometimes. There are studies that have shown that in some contexts you can, but there are many, and in many times there's differences. The differences are real because in observational studies you're guessing what confounders always to account for. And we might know about many, we might account for as many as we can, but there's always the unknown confounders that cannot be accounted for in observational research. And so randomization isn't going anywhere. It's still a really important tool to isolate the treatment effect between two groups and reduce residual bias and confounding. Lastly, I'm just going to say that we do have a vision for bringing in data, integrating health data into a learning hospital environment within Manitoba that Terry mentioned at the beginning, where we're taking we have a grant to help connect all the clinical databases in Manitoba to integrate them in a way that allows us to do research more efficiently and more inexpensively, to monitor our healthcare system, to surveil the population. And we're doing that with 3 innovative clinical trials, one that I mentioned throughout this talk called Traction in hematology and transfusion, one in oncology where we're using sorry, I mixed these up. This is the step wedge cluster trial, and Home First, which is studying home dialysis in different populations, is a non-randomized trial, an implementation effectiveness trial. We're using fairly standard statistical techniques and sort of more innovative methods of consent, and patients are engaged in every part of this process. The really unique thing about this is not only using integrated real-time clinical data, but also where we're showing benefits, partnering with the government, showing benefits that hopefully will save money, and being rewarded back or being paid back some of the savings that we demonstrate to tackle more research in Manitoba. That's a priority, and hopefully that will transform the way we do care. And we'll scale this up, we'll make it very feasible and scalable over time. So in summary, medicine has evolved. You know, when you had a heart attack 50, 70 years ago, you came in with bed rest. Bed rest is always what the doctors do when they don't know what to do. So if the doctor recommends bed rest, that's another way of saying he or she has no idea what to do. Okay? That's what we always do. And it's never of benefit, by the way. It's just never a benefit. And so now we treat heart attacks in cath labs, high-tech situation where we break up the clot or open up the artery, at least from the TASTE trial. Like there have been innovations in medicine, sometimes we haven't changed, by the way. We still call the stethoscope. We still use it. Of course, you don't see anything with a stethoscope. You hear things, so it's misnamed. It's still used despite better technologies. It still has a role, maybe, but we're not going to change that anytime soon. And there's lots of examples where we're unwilling to change in medicine, too. But we do have trials designs that are innovative. We do have statistical concepts that are advanced beyond the traditional frequentist approaches. We have methods of consent that are better and more risk adapted to certain trials at the time. And patients are now involved in the process of research and ought to be, so the results that we obtain are important to them, to family, and society. So I think you should consult clinical trial experts when thinking about trials that you're considering doing. I think you should form authentic partnerships, not only with CHI, but with the right people, whether that's government, whether that's institutional organizations, hospitals, health regions, etc., to hopefully create this learning hospital environment like we're doing, where every patient always informs the next one, where the clinical trials are part of everyday care, where research and clinical work are not separated, they're the same. So we always have the right benefits to the right patients and understand their costs and their treatment effects continually. I think that's all I have to say. Thank you very much for the opportunity to present to rounds, and I think we have time for a couple questions.
And we have a couple mics.
So you can, there's two ways to ask questions. You can go to the microphone, or you could text your question to the, that thing again.
Okay,
So fancy. I'm going to go old
So
school.
Lauren will do the in-person, and then someone will try the texting.
Yeah.
So thanks, Ryan. You know, great and happy to chat about some ways to kind of move some of these things forward. One thing I was hoping you could maybe touch upon with platform trials is the difficulties operationally, like working with multiple sponsors, if it's not necessarily an academic or investigator-led trial, and what some of the operational considerations for that might be.
Yeah. Well, out of all the trial designs I presented, these are the most avant-garde for sure. These are the most at the edge of comfort. And my group, like that I work with, only has experience now doing one. And there's like one in oncology. There's a couple others that I know about, but I don't even know their names. Like it's like that new. And we got the first phase funded. We have no idea how we're going to continue this for sure. And we like openly like acknowledge that.
So you don't have the magic solution.
We don't have the magic solution. Like how will a funder keep funding a trial that doesn't have an end?
And how do you bring in new sponsors as new products?
How do we bring a new one? So what we've done, So we've tried to fund it in pieces. We got the Canadians funding this trial, although it's embedded in many. We got the UK people funding that component. We got the Australians funding that component, and then we're hoping that we build a network and show the results of, and then go back with larger grants to Gates or Wellcome about pandemic preparedness or something that we can hook them to the next thing. But it is a challenge. I don't know what the future holds for this, but I do know this, that if you create your own future, it's easier to predict it. And so we're going to try to create this future for a platform trial that has clinical care alongside research, and have that the standard of care for clinical research and clinical trials.
Is there any text question, or do we go to the people? And then we'll go to you. We have to use this new technology.
Is this presentation available online? Oh, I think it's being live streamed right now. Slides will be posted. So yes is the answer, and then maybe there'll be an email about that, but it's on YouTube, I think. I regret that already. How receptive are funders been? Oh yeah, well I talked about that.
Control the money at CHR for trials?
I happen to be the chair of the Randomized Control Trial Committee, which I'm very proud of, but the truth is,
very receptive to
but the truth is the committee is still, like, the committee is not as innovative as I hope they would be, and these new trials are a challenge to talk about at a large group meeting. But we will get there because we have to get there.
Yes.
Like we have to. The challenges presented to us with standard trial designs, methods of consent, statistical theories are overwhelming, and change is required.
So,
Deeping.
Ryan, so before I very informative presentation on clinical trials, so before I ask a question related to your presentation, so I know you, before you travel to China, you asked me advice whether you should travel to China, whether you got in trouble to put in a jail, or something like that.
Yes,
so what's your impression
Yeah, well,
after you make the travel to China?
Well, thank you for encouraging me to go. I'm really glad I went. I was exposed to incredible colleagues. And it was a 10 out of 10 experience. They treated me so incredibly that if my wife knew that, she would send them an email saying, why did you even treat him that way? But there's so much talent and ideas and knowledge in China that has not been able to spread yet, and I was really grateful to be exposed to that.
Okay, thank you.
Yeah.
So now I ask a question related to your presentation. So you
Sure.
sorry, you're talking about p-values. So I have been spend more than 10 years teaching students about the p-values. So you have conducted so many trials. Are you looking for p-value less than 0.05 when you do these trials? We have a lot of discussion on the p-values in a high-impact journal. So some journals, a lot of scientists suggest we should raise the p-value because the force in that discovery rate, raise the p-value from 0.05 to 0.005. Some even say abandon the use of the p-value. What's your thoughts?
Well, I think you would be better to answer that than me, but I think the p-value is...
But you are conduct so many trials. Are you looking for p-value less than 0.05, or you never...
I think p-values have no place in medicine, and they haven't for many years, and they're of historical interest, but it's hard to change practice. Confidence limits, first of all, are better. Changing the p-value is a band-aid, but certainly better than the 0.05. Moving to Bayesian statistical theory is probably the answer, but we just feel it's too intimidating at the moment. Already the p-values are like 10 to the negative 9 and 11 for genomic stuff, but again, that's just, I feel like it's a band-aid. And I think we need to be bold enough to move this needle in a real substantial way.
Thank you.
Yeah.
I think the Bayesian probability curve says we should now close.
Yeah, okay.
But I want to express great thanks to you for a great round and getting us stimulated and thinking about how to do things in a new way. So thank you very much.
Well, thanks for the opportunity, yeah. It was a little bit long, sorry.